Dear Professor Gizem Dinler and esteemed participants, a warm welcome to Bioinfocongress VII. We are truly honored to have Professor Gizem Dinler with us today, presenting on targeting protein-protein interfaces for therapeutic intervention in cancer signaling pathways, an area of profound importance and intrigue. Following the presentation, we'll have an engaging Q&A session where we'll address both questions submitted by our participants and those thoughtfully prepared by our team. We look forward to everyone's participation to enrich dialogue even further. Professor Dinler, welcome once again.
Hi, hello. Can you hear me?
Yes, we can hear you.
Perfect. It's very nice to be here. Actually, I'm really honored and I'm thankful to you for this great opportunity. So today's talk gonna be about targeting protein-protein interactions for therapeutic intervention in cancer. I'm from Istanbul Technical University, from Molecular Biology and Genetics Department. Here's our campus. You can see it. We have a very lovely campus. One day we're, like if you have time, please visit us. So I pinpointed here is the MOPGAM. This is our building. I don't know whether you can see my cursor, but can you see the cursor? Hopefully. So this is MOPGAM building in green, and we have a DNA structure here. It's made wrong, but the bonding interactions, they're not that correct, but oh well, still we have a DNA structure right in the building. So this is a really lovely place. I hope one day you'll visit us. I've been using this slide for over maybe 15 or 16 years. It's really sad to say it, but it's extremely important for me, this figure. This is a cartoon representation from David Goodsell's webpage. Now he became a really big figure in the area. So what he's been doing is that he's using the cell and mimicking the cellular environment to show its, all these structures, how it's being formed. So I love it. And the reason why I love it is because it's showing us how the cellular environment is crowded. You can see here ribosome in big red blobs, and here you're seeing mRNA on white, like this string-like motif. So here translation process is taking place, and in this crowded environment, proteins has to be synthesized, and all the reactions has to go correct in order to, you know, achieve the final and functional version of the protein structures. And these functional proteins later on interact with each other to perform their function. Not necessarily they interact with each other, they can interact with other molecules like you know, RNAs, DNAs, all these different types, or lipid bilayers, in order to perform a function. And especially when a signal comes to the cell, all these interactions are being rearranged, and they're being regulated somehow. So what we're extremely interested in the lab is we think that in, you know, disease condition, this can be cancer, this can be any other type of disease condition, these interaction networks are being altered, and the alterations cause at the end of the day a disease form of the cell. So in order to understand in depth, especially cancer models, they're really for us to understand the importance and the function of all these protein-protein interactions is extremely critical, because cellular signaling pathways are so critical in cancer. And what happens, especially during treatment, most of the treatments are directed to the cellular membranes, and once the signal comes to the cellular membrane, it's sometimes you know, due to the therapies, it can be blocked. All these, you know, the mechanisms that regulate downstream pathways, those cascades, can be blocked by targeting membrane transporters or that type of protein structures. But what happens later on is that a resistance occurs for these particular drugs. This happens to antibodies, even like antibody therapies. So the idea for us when we initiated our work is that in order to either find out early detection markers or therapeutic targets in cancer specifically, especially in the early prediction of these types of diseases, we believe that there are existing molecular hubs. Like I always, you know, think that they look like these metro stations. Think about Gayrettepe metro station. Gayrettepe, there are these lines that come together, and then all these roads, they just engage in one particular station, and then it's being distributed to different roads, right? So this is the same idea when we think about cells. So there are these intersection points in the cells where we believe that targeting those intersection hubs, we can block the signal that arrives within the cell by targeting these molecular hubs. So that was the initial drive for me specifically when I initiated my lab like 16 years ago at ITU. This was the idea, and I've been still using the same slides since then, in particularly this part, because still we have the same aim. But today what happened is that after 15 years of work, we have found one molecular hub and we targeted it, and it was successful. So what I will be doing today is I'm going to be introducing how we determined that molecular hub in the cell, and then how we targeted it and how we come up with a potential drug that can be used with or in parallel. I mean, sometimes, you know, together it can be combined to other molecules that, you know, that target cancer cells, or it can be used alone for therapeutic purposes. So that's what we believe, actually. So so this was the drive, and now today, I mean, it's really fantastic, because I would never imagine that we could come to this stage, you know. So we started with asking, of course, how we can tackle this problem. And since you all know that cancer is a balance between cell survival and apoptosis, and once this balance is tipped off towards cell survival, then you know, we have like all these cell proliferations and cell divisions that cannot be controlled. And in between this two very elegantly, very fine-tuned, equilibrated balance, we come up with this protein homeostasis related solutions, and they are all these chaperone type of proteins and other proteins that are staying at the you know, the top position here. So we thought, looking at this protein homeostasis related proteins and use them as baits to find out molecular hubs would be one solution, because we think that adapter-like proteins, if we can you know, dissect out one of those proteins, and if we can, from the cell crowded environment, if we can, you know, pull out a molecular hub through the adapter protein when other proteins are bound, would be one way of detecting the molecular hub. So this was the idea. And let me introduce you first who is BAG1. BAG1 is BCL2 athanogene. It means that it binds to BCL2 at certain stages, but it's also you know, in literature there are so many examples of BAG1 having interactions with these critical proteins like CRAF, HSP70, but all the information at the time when we initiated the work was all about single interactions, like either with CRAF, direct interactions, BCL to the interaction surface wasn't deduced, CRAF interaction surface wasn't deduced at the time, and also other interaction surfaces. Only HSP70 interaction was known, but the rest, nothing was known. And this was an interesting protein because not only it functions as an adapter protein, it's involved with different pathways in the same time. You know, while it's found in protein homeostasis, it's found in anti-apoptotic functions, and it's found in cell survival related functions, and also some other pathways it's also involved in, which I just didn't want to go into. But this was a critical protein. That's my ultimate point. So let me introduce the protein to you. It's also interesting in terms of how it's being translated. It's translated starting from different initiation translation sites. Since this is the, you know, the way it's being translated, we end up having three different isoforms of the same protein having differences in the N-terminal part of the protein. As you can see on the N-terminal side, what we're seeing is that, for example, the large isoform, it's called large, medium, small. Large isoform has nuclear localization signal that targets it to the nucleus. We have in the medium some part of the nuclear localization signal, and medium isoform is also can function in the nucleus. Small isoform, whereas usually is found in the cytoplasmic environment. As you can see, all of these three isoforms have common ubiquitin-like domains and bag domains, and all the shenanigans, so-called, happen through the binding of either ubiquitin-like domain or bag domain. Especially bag domain is so interesting. There is one crystal structure available with bag domain to HSP70, and it's very obvious how the binding occurs there. And now our recent work also showed us that most of the interactions occur through the bag domain to other interacting partners. But some interactions can happen through the ubiquitin-like domain in our search also. So, okay, so now I try to explain this bag one, but what I like to do also is talk about its functional roles. It's also in some of the cancer types expressed more, and some of them they're expressed less. So it was so bizarre to me, but in most of the cancer types, we found it being expressed more. So we picked the ones, especially we picked breast cancer. At the time, my aunt, she got a breast cancer, and I was very curious about like how we can, understand its detailed signaling pathways, how we can find therapeutic targets and source of the things that I was asking at the time. And this was just coinciding with that. And in order to find out the interacting partners later on, what we had done, we had followed these like list of work that I'm showing you right here. So first of all, what we had done was, what we wanted, as I mentioned to you, we wanted to pull out all these proteins that is composing the molecular hub in the cell, in the cytoplasmic environment. So we use this bag one as a bait. So now you're going to be seeing it much better in this figure. So we use bag one, and then it has these interacting partners, but there's not only one bag 1s, there's M and L. So once we constructed bag one, since it's expressed from one messenger RNA, all these isoforms, we pulled out together as a complex. So this is the way we, from the breast cancer cells, we pull out all these complexes. Like it's like exactly like fishing. But the important thing here is that these complexes, it's really hard to maintain and maintain the structure of the complexes. It's not easy to take them intact and find out from especially mammalian cells like breast cancer cells. So it's not like bacterial work. It's not like just only one protein being pulled out. It's a difficult task. That's why it took us like how many years? Maybe 5 years to establish this path. Okay, so it wasn't easy. So let me go back and tell you the what we had done in sequentially. After once we found out like the molecular hubs, we needed to understand who they are composed of. So we after like purifying these complexes, we found out all these proteins that are constituting these complexes. And then later on, we cloned them specifically and then purified them, characterized them, and reconstituted them. And what we wanted to understand here, again, once we reconstitute the proteins that are found in the complexes, we wanted to find out the surfaces of interaction. And we used their atomic level type of studies, like where we're looking at angstrom length distant apart interactions. So the way we employ is that type of structures is through hydrogen deuterium exchange mass spec, cross-linking mass spec. We also use X-ray crystallography, cryo-EM, all these types of other structural methods as well. But these methods that I'm pointing out here, they are mass spectrometry related methods. And then in order to test the surfaces that we found that are correct, we had to introduce mutations to those particular sites, and then do lots of different types of work. Again, you can do in silico related work, and also you can do again purification and doing in vitro pull down experiments or HDX experiments again, or in vitro cellular studies you can employ. So this is the work line we followed, and I'll be explaining all these things, and I will be showing you the results related with it. So I showed you this figure. What we had done, we pulled out these complexes from breast cancer cells, okay? And then what we had done, we had to one by one identify who those interacting partners are. So this is the method. We just pulled it out, washed it out, cut it out into little pieces, and then send it to mass spec. So this is the, we had this set up in-house. So this paper is published, it's out. So that's why I didn't want to go into the details, but I'll show you the final results. How we found out exact interaction partners for each isoform. Specifically, we dissected out isoform specific interactions by this work. So you can read the paper there. And we are very proud that we have a well-equipped state-of-the-art laboratory at ITU. We have established one of the, I think the only mass spec related structural biology studies in Turkey. So we're proud for that. And we're like, our papers are coming out day by day, and we're improving the quality of the papers. That's also something, I think. It's not easy to publish in good quality journals from our country, unfortunately. So, but we can do it. That's why I think you should visit ITU at one day. So the result is BAG1S has this many number of proteins that are interacting. So each one, this short version of the IDs of these proteins, IDs of the proteins are listed. So 100 and something number of BAG1S interacting partners exist. BAG1 and M interacting partners are listed here, and L are here. So, since it's in the paper, I'm not going into details. So this was so crazy. I didn't believe in the data in the beginning. I said, like, okay, I know that this is an adapter protein. This can interact with different proteins. I know that, but this is too much, I thought. But then later on, I realized it could be possible. But then there's again downside of it, is which, you know, it would take my life or more than my life probably to express each of these proteins, purify them, characterize them, look at the interactions. It's almost impossible. So what we had done was we had looked at first the known interaction partners that literature you know, suggested that RAF1 or CRAF, they're the same protein. So that's one interaction partner, and it's a transient interaction. So we thought that that would be a nice start. And since it's important for cancer, we initiated our work from CRAF. Okay, so today's subject is going to be on CRAF. But for example, we took VCP. This is a novel interacting partner, and it's a very interesting protein. It is found in endoplasmic reticulum associated degradation, ERAD. So we have you know, done a lot of work on that too. VCP is an interactor for all the isoforms, not just one. Like here, there's also this one interacts with VCP and the other one. But today, I'm not going to be talking about VCP interactions, okay? Just CRAF, because we found something really interesting there. We also have very nice data on VCP, which I can talk later on. Maybe not today. So, okay, what after finding all these different partners, we needed to understand how do they combine. Like, it's like solving a puzzle, you know, like how are they interacting directly or indirectly? The only way you can understand it by designing these constructs and purifying them and looking at their interactions later on, whether that's the only way to understand it. So we, in order to you know, do that, we had to purify the proteins separately this time, not as a complex, separately. Okay, so since I'm going to be talking about BAG1 and CRAF today, I will concentrate on these two proteins. Okay. But then, as I told you, we had really nice data on VCP-P97 interaction with BAG1 and BAG1 with BCL2 and HSP70. Now we're trying to put everything together in this big puzzle. But one thing that I skipped to tell you guys is that now this is just a static view, but you have to be always thinking about the cell as a very dynamic, very, you know, instantly changing environment. So these interactions in time change depending on the signal that's been received. Therefore, it's a big question. So now we're trying to understand it in cancer environment, but we needed to be understanding the interactions in a different type of a setting, like, you know, autophagic environment, for example, or in healthy environment, in order to deduce the differential interaction platform. Okay, so this is the final aim for us, but we're not there yet. So let's come to today's talk, which is on the CRAF interactions of BAG1. Therefore, I like to introduce CRAF, who that person is. That's a protein also. It's a MAP kinase found right below ROS. So it receives signals from ROS, and then it modulates this MAP kinase cascade by recruiting MEK and ERK, and then that ERK is transferred to nucleus, and it recruits all these cell survival related genes. So it's a very critical pathway, but this CRAF is a very interesting protein by itself. So my student Özge, now she's also a professor at Medeniyet University. She is she has come on, I mean, she went crazy purifying this protein. It's because it's not a stable protein. It has these unstructured regions, and it makes it prone for the degradation, either through proteases. It gets digested, and then we don't get a stable protein at the end of the day. Özge, for that reason, she started working in mammalian cells, trying to produce CRAF through mammalian cells. She wasn't successful. She tried bacterial cells. She tried different versions of it. And then I said, okay, we're a protein lab. I know Özge. I thought she's going crazy. That's why I said, okay, let's buy the protein from a source, different source. Maybe we couldn't do it, you know. Okay, we can live with it, I said. And then we bought the protein, and it wasn't functional either. It was all chopped off into little pieces again. So therefore, we decided, okay, what can we do? Then she found a way to purify the protein as a in the form of a full-length structure, and also she designed these shorter versions, truncated versions of CRAF, and she found her way, but it took her 3 years to purify this protein and characterize it. But since it's a very interesting protein, it's a kinase, and RUS signaling pathway, RUS mutations cause 30% of the known cancer types on Earth. So it's a very big you know, pool of cancer types. It constitutes you know, like 1/3 of the people that you see having cancer can have these RUS mutations. So it's found in pancreatic cells, colorectal cells, lung, and melanoma. And the way it's being the way it functions as an oncogene is taking these this MAP kinase cascade in action. So it's very important, and it's dependent on CRAF. Okay, so that's why these RUS mutated versions of the cells are critical for us to understand. So we thought we can, if we find the surface of interaction with BAG1S and CRAF, we said this can be one therapeutic option. Okay, this was the objective. So in order to do that, what Özge did, she had to find the druggable sites on the protein BAG1. By the way, BAG1 structure wasn't available at the time. For humans, still it's not known. For some other species, it's known, but we don't have the full-length structure of the protein in the X-ray form. Neither cryo-EM. We have the domain structures available, so it's not easy to find out the druggable sites. I have to tell you that. And the second step is to identify, detect the potential interaction surfaces between these two proteins. And the third thing is to find out the hotspots on this interaction surfaces, and then find and design a molecule that can inhibit that interaction. And we use peptide-based inhibitors. So we designed a peptide-based inhibitor to target these surfaces. So this is the story that I'm going to be talking about today. So the first thing that one needs to find is the druggable sites on proteins, and it's not an easy task. Some proteins have these very how can I say, very deep clefts in while binding ligands or interaction partners, but some proteins, they have these shallow surfaces. They don't have very distinct surfaces for binding. this means that they can bind a variety of molecules. So in order to find these shallow surfaces for drug purposing, it's not easy. So this is the task we had to tackle with in the first place. So for that, we need structural data. We needed to have structural data. That's why we employed all these structural tools. Okay. So the first thing we had to do is we have to purify the proteins in their own sources. Since we are working with breast cancer cells, we used breast cancer cells for the protein expressions, and it wasn't also an easy thing to purify a protein from mammalian cells, and we didn't have really humongous amounts. So by the way, it got accepted just recently, 3 weeks ago. So it's it's already printed. Sorry, I just wrote here in print, but you can read the paper. It's a really nice story. I loved it. I hope you like it too. So this is the way she designed purification. She had the construct like that, having the his tag, but we always remove the tags. We believe that tags interfere with interactions. And then later on, what we do is we characterize the proteins well, whether they are, purified, as a intact form. In the the purity is important for us, and we think that purity should be over 90% in order to pursue with binding experiment. So we had all this protein having, like, we had the his tagged version of BAG1S, and now we cleaved off the tag and we got rid of the his tag, and it's beautifully purified and characterized well. We use size exclusion chromatography to analyze the stoichiometry in solution, and we found out that it's a monomeric protein. It's found in the monomer form, this peak, as you can see, and its secondary structure is showing a mostly alpha helical folded protein. So it means that it is folded when we purified it. So it's these things we say, okay, tick, tick, tick. We have a pure protein that we can further do binding experiments with. But then we also wanted to understand how these proteins behave in solution, and how these molecules interact with each other. So this technique that I'm going to be introducing to you is a very, very powerful technique, and it's been used worldwide. I know especially in pharmaceutical companies, these techniques are used a lot. Not in Turkey much yet, but I hope it one day. But in Roche and Pfizer, they are using it very often. So it's called hydrogen deuterium exchange mass spectrometry. Here you are seeing Baran running the experiment. So the technique is very simple. The idea is very simple. What we do is we take advantage of the difference in between the isotopes in mass. Their difference is critical to us. That's why we're using mass spectrometry in the first place. And what we do is, now I'm going to introducing to you a concept. Maybe you already know it, but if you don't, molecules breathe in water. So water for us is extremely critical. All these molecules, the breathing motions happen, and we can never obviously see or detect that in water, especially with molecules having these hydrogen bonds, proton sites that are exchangeable, labile, they constantly exchange with water protons. They change places. We cannot see it. We can never see it. This is called proton transfer, and this has always been happening. And in our body, this has been happening too. And this causes like all these vibrational and rotational motions. And you know, at the end of the day, what happens is that molecules breathe, we call. And this breathing motion for like each proton site has a different rate in exchange. Okay, so if a protein is folded, the protons that lie within the core of the protein would exchange slower as opposed to the protons that are solvent exposed. So there's gonna be a rate difference in the exchange process. Okay, this is so critical. Please try to remember it. Okay, so we took advantage of this difference. Okay, so the idea is, as you can see, we have the protein in water in this form, and then we take it out, and then instead of water, we put deuterated water, the heavy water, and then the hydrogens that are solvent exposed would exchange rapidly with the deuterium deuterons. The ones that are protected from these, you know, from the solvent, they would be exchanging only when they breathe, when they have these vibrational motions. This allows us to see all these dynamics that are taking place in our protein. So this is extremely critical. Okay. This technique not only finds out these conformational rearrangements or allosteric conformational changes in proteins, it can also tell us the interaction surfaces. Like, for example, here this slide is very, you know, very beautifully explaining, I think, this fact. Here what you're seeing is that the protein, when it's by itself, this particular site is solvent exposed. But once this site is being protected by the binding of another protein or another molecule, ligand, DNA, RNA, doesn't matter what it is, but it won't be exchanging as fast. Now we can find out where exactly this interaction is occurring. Okay, this is the way we do our experiments, and that's what Özge did. So this is, you're seeing Özge. So this is the sequence of the protein. We have these peptides. The way we do the experiments is we have to first cover so let me tell you this, the experimental wise, what we do is we have the protein, and then in water we put it in deuterated water, and then we stop the reaction, and then we use pepsin to digest our proteins into little pieces. So we have the peptides in hand for that particular protein. So we have now switch to peptides. We identify in mass spec peptides, and we identify the peptides when they are not deuterated and once they are deuterated. Okay, once they are getting deuterated in time, we see a profile of deuteration, and each deuterium addition would bring one atomic mass unit difference. So we detect the shifts in the peptides by looking at the deuteration level. Okay, we know the peptide sequences. So this is exactly the final final result. What we see here is that, as you see on the y-axis, the time axis, on the x-axis you're seeing the sequence of the protein, but each time point you're seeing differences in the color codes. Okay, so this is called a heat map. As you know, in this heat map, what we look at is the regions on the protein that are exchanging in a slower pace, because the ones that are exchanging in slower pace would tell us those are more protected regions, especially when you saw 0 here in the scale of the color scaling. The bluish color is telling us that it's a protected site away from water. Okay, so they would exchange later in a slower pace, and the ones that are solvent exposed would exchange very rapidly, and they're in red. Okay, so you're seeing a difference in the scale in the form of the exchange rate. So especially Özge found out this blue part as the protected site, and she postulated that this would be the druggable site on the protein because it's being protected from solvent and it's exchanging in a slower way with deuterium. So once she mapped out all the findings on a time scale to the structure that we found out with AlphaFold, this is the first structure when it's not deuterated, and in time, as you see, it's being deuterated in 24 hours. What happens? You're seeing it here, and you start seeing these cyan color protected regions, and she found out that this part especially is where is the site for probable interaction. So this is what she found out. These results I always show, but the only reason why I like to show it is because in the paper you're going to be reading it, if you will, but telling you all the peptides, how they acquire hydrogens in time. So this is what has been shown, how we make the experiment. So here what you're seeing is each plot has demonstrating us the peptide sequence, and these plots in red, what you're seeing is the as a function of time, how much proton is being becoming deuterium instead of hydrogen. So we are looking at incorporation of deuterium in time. Okay, so we are seeing a change, and as you can see, some of these sites are exchanging with kinetics, but some of them stay as it is. So those sites, they're already exchanged, and they're saturated. They're not getting more hydrogens. This is what it means. So these are the details I can tell you, because these are so critical. The way we analyze data is something like that. There are two types of exchange mechanisms. One exchange mechanism is if a protein is already unfolded, it exchanges rapidly. But if a protein is how can I say, folded, it will exchange in time in a slower pace. So in EX1 mechanism, we are seeing a bimodal distribution where this demonstrates us it's a rapid change. It's an unfolded part of the protein. We can say that. On the EX2 mechanism, it's the native-like structures resemble in this type of an exchange mechanism. So we deduce the results like that. And also we look at the data. So each here we're seeing in time each peptides how they acquire these protons exchanging with deuteriums in time. So this is what we are showing here. It's an example of bimodal distribution. Some peptides are showing that type of an exchange rate, but some of them are showing EX2 type of a mechanism. So EX1s, as I told you, are more unstructured region related part of the protein. So we looked at the same thing for bag domain, and we have all these graphs, distributions. We have, again, both bimodal and regular native-like distributions, EX1 and EX2 mechanisms. We figured that out. And then once we mapped it on on the structure, this is how it looks like on the structure. This cyan color part of the protein is the druggable site, as we determined. Okay, so this is the structure, and it's now we have a dynamic behavior of our protein, okay, in details. So these are my students. Mirai is finishing now. She's going to Germany for postdoc. Cemre is doing her master's, almost completing it soon, I hope. They're explaining how they run the experiment in the lab. Mirai, she's taking her PhD. She had done an extremely amazing job. She has an unbelievable story. Maybe one day I'll tell you that story as well. It's about BCL2 and pectin-1 interaction. She found new sites for drug targeting. It's beautiful work. Its paper is gonna be out soon, but maybe I can talk about it also again, because it's about glioblastoma, and that's a terminally, it's a horrible disease. Anyways, so let me skip that because I already explained it. As you can see, we found out how our BAG1 is behaving, and now we needed to do the same thing with CRAF and find out the CRAF interaction surface, and then find out the hotspots and everything. So in order to find out the interaction surfaces, we first had to, of course, find out whether they are really interacting in cell. I know there's a lot of literature information, but still we always do it in our hands, whether we obtain the same results. Exactly, they're interacting. We did a lot of pull down experiments, his pull down, tap pull down. We use different tags to pull down the proteins together, and we see them. They're coming along together, they're interacting. This is good. This has been known. This wasn't something new, but we are like keen on doing all these experiments to see whether that's the case. And then we did cross-linking experiments. We use a cross-linker called DSS, this molecule. We link these two proteins together. It's like a glue. We glued them, and if they're in a close proximity, they can get glued. Otherwise, this cross-linker doesn't work. And it works. As you can see, these two proteins, once we glue them, they're being glued. Okay, they're together. And in order to find out the interaction surfaces, as I told you, atomic resolution models we use in order to determine the interaction surfaces. Mass spec, we use computational studies, all of these together. But the problem here arose with CRAF. CRAF, once we had to purify it, it was a very difficult protein to purify. So it because of its low stability and low yield, it was a big problem. And the way Özge tackled this problem, what she did was this experiment is very beautiful experiment. I urge you, one day if you're looking at interactions of two proteins, you can use this. But always just keep in mind, please, when you are looking at protein-protein interactions, you have to always employ orthogonal methods. One method only cannot tell you exact results. So that's very critical. So what she had done, she used a membrane. She purified one protein and put it on the membrane, immobilized it on the membrane, and then ran the other protein on top. Okay, let them interact, and then she did proteolysis. She cut the protein out, like the ones that are sticking out, not binding, and then she washed away the ones that are sticking on the bound protein. okay, would stick on the membrane. Okay, the rest would be washed away. So she scraped it off from the membrane and digested it, and then did MSMS, mass spec analysis, and then what she had found was these two proteins in the membrane, they're binding. And also what she had shown here is that the catalytical CRAF domain, because she had these other versions, truncated versions, as I told you, catalytical domain itself is present in her experiments, they're always binding. So what she said was they're binding from the catalytical domain. And then next, she found out from that MSMS study where exactly these two proteins are interacting. So these sequences that I'm showing you here on the slide is pointing out where exactly that these two proteins are interacting. As you see from the this pink sides, these two proteins are interacting with each other. Okay, so that's what we found. Then we always use in silico methods in parallel to help our research. The beauty about this is that we first found out the interaction surface using experimental methods, and then we employ in silico methods. So they're like, we are using it in order to, you know, fine tune which residue exactly is in the contact with the neighboring interacting partner. Okay, so now we found out several candidates for interaction by doing this HEDOC dock. We had like multiple docking sites, different poses, and then from those poses we had this number of possible mutations that we can substitute to bag one. So now we go back to experimental study again, and then we had introduced these mutations to the protein, and then again we had purified it, characterized these proteins, and then did a lot of different types of experiments. The first experiment we had done was we used this time in vitro systems, and we looked at the signaling pathways, how like introduction of these point mutations would alter signaling cascades. Okay, since BAG1 interacts with CRAF, and CRAF gets upon it gets phosphorylated, it goes and phosphorylates MEC. Okay, and then MEC phosphorylation recruits also ERK, and then that's the MAP kinase signaling pathway, how it functions. So once we look at the phosphoforms of these proteins, we are looking at the how the activity is being altered depending on the mutation. So as you see here in the box one, the activity of these proteins get lowered once you look at the phosphoforms of the proteins. So we said, okay, now 1 point mutation affect the cellular environment, and then we looked at the other point mutations, and we see similar effects. The phosphorylation levels change for these proteins. So this is important and critical. You're introducing 1 point mutation on a protein, and then it changes the partner protein signaling ability. This is so critical. So that was something. So we looked at different sites, as I told you, and we thought they might be the hotspots. And then we had done all these, classical, very, well-known molecular biology experiments. Here you're seeing MTT. It's a WST experiment, sorry, not MTT, but we looked at cell viability here, and then we see a change in cell viability. So once we introduce these point mutations, cancer progression slowed down. So this was something, I think. So then we also looked at, since this is an adapter protein and deducing out the interaction site, the determining the hotspot would allow us to look at other partner proteins, whether those other partner proteins would affect it or not by CRAF interaction blockage of BAG1S, because we know that BAG1S has interaction with Beclin-1 and AKT and BRAF somehow, but these are not well known whether they're direct or indirect. But at the end of the day, point mutations affected those interactions as well. So this was something, especially BRAF, Beclin-1, and AKT. We used HSP70. Even HSP70 interactions has, a little bit, the levels are very, like, this is not a quantitative experiment, but still pull down experiments semi-quantitatively tell us that it's lowered. BCL2 interactions got altered also. So these are critical. And then we ask this question, do these interactions occur from the same region or are they mediated by CRAF? So this is one way to understand whether CRAF is a mediator of these interactions. BAG1S doesn't directly interact with BRAF or Beclin-1. That's what we found out with this study. This is the experimental design, and we show that once CRAF is removed, there is no binding with Beclin-1 nor BRAF. So, but it still binds HSP70. It's a direct binder. We knew that. So this was also very, for us, it was very informative and it was very nice to see this result. So this is the way we think these processes occur. So then another thing came up, which is whether we can disrupt these interaction by introducing a peptide that either binds to one of these proteins and block the interaction. Since we know where these two proteins are interacting, we can use the sites of interaction as an inhibitor for one of those proteins. For example, we can take BAG1S peptide, one part that interacts with CRAF, and use it as an inhibitor to disrupt this interaction, because they will both BAG1S and the peptide that has the binding site would compete for the binding site on CRAF, for example, or vice versa. So this was the idea. We designed 4 peptides for that. These are the peptides. 2 of them target CRAF, 2 of them targets BAG1. We purified them, characterized them after synthesis. Boom, one of them worked. Peptide 3, PEP3 worked. Okay, so we thought this can have a potential to be an inhibitor. So we did a lot of experiments to see their interaction propensity with the surface for inhibition. And as you see, when we have these 2 proteins in hand, and then we introduce PEP3, this is the control peptide, it's not affecting the interaction. When we introduce PEP3, it blocked, as you can see. So there's no interaction. We were so happy. I cannot explain how happy we were. And so it also affected other protein binding interactions. So this PEP3 has a big potential, we thought. It's a blockage. It's blocking BAG1S interactions with other proteins, not just CRAF, obviously. Since it's an adapter protein and we determined the hotspot, So this is something we thought. And then, of course, now we know that this peptide, we designed it, but it has to penetrate through the membrane and reach to the cytoplasm in order to function. So we had a penetratable version. We designed the penetratable version of this peptide. It could go into the cytoplasm, crossing the membrane. And then since this became a new peptide, not any longer the same peptide, we had to look at its all potential and binding and all sorts of things. So that's what we had done here. We, did those experiments, binding of PEP3 only. This is the penetratable version of it. We applied for the grant, patent, sorry. And then we had looked at the binding kinetics of these two peptides. So the binding affinity here for this is PEP3 binding affinity. We looked at the affinity for GoPep. We named our membrane crossing version of it as GoPep from Gizem and Özge's names, initials, GoPeptide. GoPep. Now our GoPep is working. It's going along. It's a good amount of binding affinity. We liked it. So as a conclusion, in order to sum up, we determined the druggable site on the protein. We looked at the interaction surface. We determined the interaction surface of these two proteins. We determined the hotspots, and we designed the peptides in order to inhibit that interaction. So then the story is never finishing. Of course, it's gonna. We had to look at its potential for being a drug. So we looked at its potential in the different in vitro cell systems. This is the colon cancer HCT 116. This is MCF7 cells. As you can see, the potential, drug potential is pretty high. So this micromolar range is a good range for inhibition purposes. So it worked in our cancer cells. We looked at regular other studies like colony formation effect of this peptide, and it's getting lowered upon, as you can see, it stops cancer formation. It's a really good finding, I believe. We did, as you can see, we looked at using flow cytometry. We looked at its dose dependency. What's the dose? We checked it by using that on MCF7 cells. And in order to understand the lethal dosage on the healthy cells, we used two different cell lines this time. This one is Beas 2B. This is lung cancer, and the hex cells. So it's not in the same range. So it's pretty high, 10 times higher. So it's not causing too much damage on the healthy cells. Not much. I mean, we're not in the high dosage range, so it's okay. Now we're in the animal testing. We had done some zebrafish experiments, and on a melanoma model, it worked also on RAS mutant cells that we used, and then obtained the model, and then it worked. So it's very promising, and we have this project that got funded, and now we are looking for partners to work on this project. If you're interested, please write me an email. Now we're in the animal testing stage. Hopefully it works, and then we'll have a potential drug for clinical therapies. I feel like once this project, I mean, at the end, maybe it's it will be successful, then I thought we found the cancer's aspirin. I don't know. We'll see. We'll see in time what's gonna happen. I always show you this slide because I want you to see our path. It's been for me so many years of work and tears, blood, it wasn't easy to come to this stage. And in the meantime, I'm an entrepreneur at the same time, and I like to urge everyone, if you're interested in coming up with something else, designing something for mankind use, I think it's very, very important, and I support it. I try my best to be an example in that. So we sell proteins in our spin-off. Horizon Biosolutions. The other one I wasn't successful with. GDD Biolabs failed, but Horizon Biosolutions still functional, not bad. So we try our best to perform there. So this is the crew, current crew. We have so many graduates, alumni, which I am very thankful of, and I like to thank all my students. They're all wonderful. I love them. And the funding sources, we are very, very, lucky to have all these grants in these years. Otherwise, this could never happen. So thank you for listening. I am good with timing, I think. Not bad. So I can answer all your questions. Thank you. And there's this upcoming conference, TUPA conference. We like to, this is at ITU. Please sign up for that.
Professor Dinler Doğanay, thank you sincerely for this enlightening presentation. We are unable to relay questions from our participants as it is time for the next talk. However, participants can reach out to you via email for their questions. Thank you very much for taking the time to share your knowledge and for your informative presentation. On behalf of myself, our team, and all the participants.
Thank you. Thank you very much. I'm sorry for this delay a little bit. I wasn't very, good at timing then. Sorry.
Thank you very much. We wish you all the best for your health and success in your studies.
Thank you. Okay. Me too.